I will do some sharing, and you've got it on record, I'm hoping.
I believe I do.
Now, the only difference is I've had troubles with sharing the entire screen, and it generally doesn't like it, but it looks like it's accepting it. So there we are. We have an entire screen shared. So, okay. First, I got a phone call during last week sometime from somebody who said, I want to ask a stupid question. Now, what's a stupid question? Does anyone know? No, I don't know whether anyone knows what the stupid question was, but what is a stupid question?
No such thing, is there?
I wonder. A stupid question would be a question that was asked with somebody not really wanting an answer. And I'm not talking about a rhetorical question, but a question where, you know, it's just plain pointless and dumb to ask. So that was the question that was asked, can I ask you a stupid question? And the question was, what is SAP? That's a really interesting question. So it wasn't a silly question at all. It was actually quite a good question. And I'll attribute the asker was Peter Gray, just in case you're wondering. So I guess the question is, Peter aside, what is SAP?
I guess it's a makeup of water and nutrients and elements inside the tree.
Yeah, that's a pretty good definition. The answer is it's like what is a tree. It isn't a real definition because it encompasses lots of things. So a tree could include bamboo. It could include a dicotyledonous woody plant. It could include a palm. It could include bamboo. All of those are trees because there isn't really a definition of a tree. That's what we make it. And if we look at it in terms of groupings, we get usually you say dicotyledonous trees. So woody dicotyledonous, taller, bigger plants, trees. We say, oh, but monocots aren't because they don't undergo secondary thickening, except some of them do. Are they trees? And gymnosperms, they're really not angiosperms, so they're in a different group altogether. How do we lasso two separate groups together? And so the definition of a tree is just an artificial construct. We can have it whatever we want, and I think you know by now, or hopefully you do, my definition of a tree is anything that's woody, woody enough to beat a person to death with. That makes it a tree. Okay, so bamboo, palms, all of those fit into that category. But a banana palm is not a palm, it's actually a herb. You couldn't beat a person to death with a banana palm. You'd have to suffocate them with it, okay, because it's too sloshy. So again, it's whatever you want it to be in terms of a definition, but for the broader terms, that's a tree for me. So sap is much the same. You know, I'm an old-fashioned boy. Sap to me is that sticky stuff you get out of the tree. And so you could say, well, that's our exudates. And our exudates are, well, what are they? I'm not going to tell you. You're going to tell me. Give me an exudate, just one.
Kyno
from Mutaceae. A couple of other completely unrelated families that produce, or at least one other unrelated family that produces kynos.
Can I interrupt you for a moment?
Yeah.
On my screen here, it says to avoid an infinity mirror, don't share your entire screen of browser windows. Share just a tab or different windows.
Okay. I will take that. So I'll stop presenting and come back to that. That's fine. So that's the first thing is, it's that stuff called kyno. That's an exudate. What's another exudate you can think of?
Resin.
Who said resin? Andrew, yeah, resin. Resin's a really important one. And resins fit into those volatile things, so they're often involved with terpenes. The kyno, it's involved with tannins. So you can start seeing groups of chemicals start together. Terpenes are combustible. And so resin is combustible. And that's one of the problems when we get Pinus radiata fires, for example. The green Pinus radiata burns better than semi-dried Pinus radiata, because it's got all these volatiles in it, all these terpenes. So we've got resin. Resin, by the way, is used in lots of things, so it's a very important product. What else can you think of?
Mother exudate.
Yeah, other exudates.
What about gum in wattles?
Yeah, so gum acacia. And that's a really important, that's a carbohydrate-based one now. So again, got a whole nother mechanism. Some of them have tannins, but a lot of them are quite edible. And in fact, you can eat the gum acacia out of acacia. You can eat it out of brachychiton. You know, it's just a complex carbohydrate that almost forms a mucousy type material. So, and we see that, for example, if you've ever made an agar plate, that's a similar sort of reaction. So yeah, gum. So what's another one? Anything else?
Oil.
Yeah, we do get some volatile oils, but they're usually out of the leaf, not anything else. We get a whole pile of these interesting things. So in the history of the world, for example, the resins out of Araucaria, not out of Araucaria, out of agathis, was a major export out of New Zealand. It was used, for example, in the production of linoleum. Some of you won't know what that is, but it's a material that used to go on the floor of places. A lot of our things that we use for polishes came from it. Lipsticks, for example, came from the exudate out of the dragon's blood, Dracaena draco, and the equivalent species out of Socotra, Dracaena, whatever that is, I can never remember. So you end up with a whole pile of these things that produce frankincense and myrrh. You know, we really do have this magnificent association with these chemicals, and there are some fascinating books on the subject. So that's what I traditionally see as exudates. And I would have said saps, you know, but, you know, I also knew that the sap was flowing inside the bark. And so that was a different sort of a sap. That was a sap that was made out of water and sugars. So predominantly sucrose. So sucrose, glucose is glucose is a step past the end result of photosynthesis, but it's we finish off with a glucose molecule, switch that together with another glucose molecule, not quite correct, but it'll do for the purpose of take out one water molecule, and you end up with sucrose. So sucrose is a 2 sugar. Sugar. And so we take that and we move it, and we take that and we move it up and down the tree. So it goes down to the roots, storage, comes up from the root, from the storage, goes up to flowers and fruit at the end. So if you think about a rose or a citrus, my big pomelos that are growing here at the moment, they're going to take a huge amount of sugar out of the tree to get to the very end. So it goes, sugars go up and down. So that's another sort of sap. And when I think of sap-sucking insects, that's the ones I think of, the ones that are feeding in the phloem. But of course, there's other saps just to mess with us, okay? There's the sap inside the cell, the cytosol, okay? Cytosol's its correct or technical name. That's everything inside that membrane, that phospholipid membrane of the cell. That cytosol is a sap. And so some of our sap-sucking insects, like lacebugs, for example, lacebugs feed on that sort of sap. They're a sap-sucking insect. And what we're saying there is they're sucking on the inside. And then the last group of saps is a sap that goes up and down the inside. So what we talk about is our apoplastic water, or our water that goes up and down in the tubes, okay? And so that's a sap as well. And when we talk about making maple syrup, we actually tap that sap. Okay, we drill into the xylem and we tap the fluids out of the xylem. So there really isn't a clear definition. One of the exudates we didn't talk about, Peter, that's really important, is latex. Latex is an incredibly important thing, and we know that some people can have reactions to some of the proteins in these latex group of chemicals. And so we have, for example, our latex that we generally use is from Hovia brasiliensis, not from Ficus elastica. Ficus elastica was used as an alternative to produce a slightly different latex, and that slightly different latex has different proteins and different responses. And of course, there are a whole pile of Euphorbiaceae that have these secondary chemicals in those latexes that, again, are nasty and unpleasant. And then you get things that adapt to it, like possums that can still eat the leaves on a. Darren, you've got a something going on in the background there. Just mute if you can. I don't know that I can mute you. No, I can't.
Mate, totally sorry. New computer and whatnot, and
That's cool.
I can't get it to work.
Yeah, that's fine. Just if you get the chance, then do so.
Muted, Aaron.
Peter's got it sorted. So yes, in terms of chemicals, as I said, these products are incredibly important. You know, we still use, for example, lac balsam, which is a resin for mounting microscope slides, still to this day. Some of the new synthetics are getting there, but again, they have their own unique problems. And plants, if we remember, are chemically complex. So plants produce a whole pile of things that we as people haven't been able to emulate yet experimentally or in the lab. So that's why we keep on saying, look, you know, maybe there'll be a chemical in the rainforest that will have some benefits. And who here's ever had a taste of a plant material or a taste of a medicine that's really bitter and astringent, or you just think, oh, yuck, that's horrible. You want to spit, and okay. Those are often alkaloids, and our body naturally tends to not like those. And the whole pile of alkaloids that are wonderful in medicine, and a lot of them were originally found in plants. Some of them, of course, are great for other things. But you think about it, our painkillers are still very much opiates, very much to this day, and still growing in poppy fields. It's amazing when you think about it. So plants do a lot better job in many ways than we do. Yes, Andrew.
Slime flux on an elm, is that considered an exudate, or is that different again?
Okay, it's different again. So our slime fluxes are mostly bacterial. And bacteria is good at breaking down sugars. There's sugars in the plant, in the wood, in the starches, for example. And I think, has everyone here seen what starch looks like inside a plant? No, okay. It's microscopic, small little molecules. You can see them at really high magnification. But these are small, very heavily folded bits of sugar that aren't normally available. But you can get bacteria to go in and it starts to break it down. Now, when you take bacteria and you break down a sugar without air, does anyone know what that does or what that process is? That's the process of fermentation. We're going to produce an end product, and the end product is
Alcohol.
Alcohol, yeah. So one of the things that comes out of those is this alcohol mix and a waste product. The waste product, of course, is a carbon dioxide, so it bubbles. And so you see the slime flux forcing water, fluids, and alcohol and everything else in there. So generally, alcohol we consider to be an antibiotic. That is, it kills living things. That's why, not me, but that's why Andrew drinks it, because he likes that numbing sensation that comes when a little bit of alcohol gets in the blood, gets it past the blood-brain barrier and ends up killing a few brain cells. And he says, oh, that's good. I'm not saying you have, Andrew, ever, but you might have done at some stage. So if you never have, I apologize. I never have.
Andrew's got plenty of brain cells, so he can.
Yeah, and I'm already enough an a-hole without adding alcohol, so I think that's not a good combination. But so that's slime flux. And what's really fascinating about slime flux is oftentimes you'll see insects feeding on it. Insects love alcohol. Alcohol is a molecule or is a chemical that has 7 kilocalories per gram whereas starch only has 4, protein has 4, carbohydrates and proteins, and fats have 9. So it's a really good source of energy as well, which is why you see drunk people who can still manage, without eating a meal, put on weight. That beer guts impart some of the alcohol as calories. So animals recognize this, and it's not uncommon for animals to do things that get themselves drunk. I was just watching a doco the other day, and bears, grizzly bears, do exactly the same thing in eating apples off apple trees that are getting old and fermented, and they end up with this thing. And different animals process it differently. So, for example, if you have a dog and you want to settle your dog down around fireworks night, go back 50 years ago, we had this thing that would happen once a year. Fireworks would go off everywhere, and the dog was uptight. Just give it a beer or a scotch or whatever you want, and they don't metabolize alcohol well, so it stays in their system for a long time. We do metabolize it, and of course we put some of it out in our breath and some of it out in our urine, and so it breaks down for us in a matter of a few hours, 8 hours or so, and we're getting back down to close to neutral. So different animals respond differently to that alcohol source, and some of the insects do a really great job. And if you want, I've got a little video footage. Let me see if I can share. How am I going to do this? I should be able to share a new tab now. Yeah, new tab. Share. And hopefully I can share from my photo collection. Slime Flux, it'll see if it come up. It's smart enough to pick up what I've put in. No, it's only got the one, so I have to do it another way. So explore. Yep, I can do that way. Now, probably should cut down a few of my screens, shouldn't I? Window, this PC, share. Okay, I go to by date and photos and photo sets from others. This is one that was shared with me by a student from the Botanic Gardens at Mount Annan. And I'll just show you the pictures and then the video. So not the most perfect image, and I apologize, but certainly do the job. So here we've got flies. You can see the frothiness. That's the very process we're talking about. When it's dry, David, is really great, is that the alcohol acts as a preservative. So you don't get a lot of decay associated with slime flux. You get necrosis or death of the adjacent tissue, but not decay. So decay is fairly unusual. So you can see, great attractant of insects. Now, here's the video, and you can actually see and hear it frothing. No, it won't let me share that. I've got to unshare. Stop sharing. And I've got to go back, and I've got to share. Video, windows, window, media, share. Okay. Can you see that?
Unknown caller.
Yep. Let me just grab this, guys. Hello. Okay, thanks. Bye. Yep, sorry about that. Can you guys see that video?
I can see a blank screen.
No, you don't see the video.
No, I'm not seeing it.
Nobody else?
No.
Okay, let me try one more time. Stop sharing. And then share window. Let's try a window this time. How's that? No?
Hang on. Still no.
Okay. Clearly we can't share videos. I apologize for that. So yes, but you can see it bubbling up quite vigorously and coming out. So what we often see is the slime flux after that process has gone on. The sugars have been used. The bacteria is now redundant. The alcohol has preserved it, and we just get a wound and not a cause for concern. If you get lots of it, it can be a problem. And trees that move sugars around in their xylem are perhaps more prone to it. So things like liquid amber, which is closely related in terms of its functionality to maple syrup. It's another tree you can tap to get syrup from, hence the name sweet gum. Okay. You can see it on sweet gums, and you'll get a patch after patch after patch of this bacterial slime flux, and the trees smell. That's the usual thing that gets people concerned about it. So that's SAP, and they're a great group of things, and they do a broad things because we're talking about a whole pile of different things. So the exudates tend to be protective. Obviously, our cytosol's there for the function of the cell. Our apoplastic fluids in the xylem, they're obviously important for water and solute transport. So, you know, water's taken in, goes up those tubes. And then, of course, our traditional sap in the bark of the tree goes up and down as well. So, again, we use one word for so many things and it makes it confusing. The other thing that happens in botany and in arboriculture all the time is we use multiple words for the same thing. And sometimes those words have subtle differences. And we've done it before, for example, vigor, vitality, health. You know, those are three words that tend to be used so interchangeably. Risk and safety and hazard are another group of words that, you know, somehow or other get whacked together, and they're not the same thing at all. And, you know, I often do it, and you know, I'm very alert to it. You know, I'll talk about the risk of something happening, and it's not the risk of, it's the likelihood of something happening, because the risk includes the consequence. Now, it could be the risk of a branch fouling, and that's the consequence. Therefore, we're really still saying likelihood. It's the likelihood of the branch fouling, and the likelihood of the branch fouling, and the failure of the branch is the consequence. So hopefully that makes sense. So,
hey, Mark, can I say something on that?
yes, you can.
Sorry, I don't know if it's noisy. I'm in the car.
No, you're
but,
fine. Great.
I was actually reading the development standard before, and on that with vigor and vitality, it seemed to say there that they're all the same.
Yeah. So again,
is that right? Is that how you read the standard?
that's exactly what the standard says.
Yeah.
And I know there was a proposal for that to be changed. The best of my recollection, and I sit on that committee, the best of my recollection, it was thought that was still too pedantic for our profession to adjust to those changes. But that doesn't mean that I can't do it and you can't do it. It doesn't mean that we can't be the leaders rather than waiting for everything else to change. I mean, you know, a typical thing would be, I still read a lot of publications, a lot of texts, even research papers talk about feeder roots. And yet, you know, they're not really feeder roots. They're small woody roots and small roots that take up water absorbing roots. Again, so I think our choice of words is really important in terms of how we communicate. And I think that's one of the problems we strike in communicating. When we write a report, we're trying to often communicate fairly complex things in a simple manner. And we've got to be careful when we do that we don't lose meaning. So I think, Nisay, that it's Niels, isn't it?
Yeah. Sorry.
Yeah.
Yeah.
It's okay. I think, Niels, while some people would use those words the same, then if you said to them, well, how would you describe a tree's genetic potential? Would you say that, for example, ficus has greater genetic vigor than an acacia? The answer would be yes. So clearly there they would use the word vigor, they wouldn't use the word health. They wouldn't say it's got genetic health.
They really are different
So
words.
when push comes to shove, we really do. Sorry?
They really are different words, or they all have different meanings.
Yeah, they are. Yeah. And botany is rife with it. So it's not just a boriculture. It really is a process that we need to add precision to the language. And we use some weird things like, and those of you who have gone down to the University of Melbourne or read a good anatomy or physiology text will have come across stuff called ground tissue. And you sit there and think, why would you call, short-lived parenchyma that's being used as a vegetable sponge, why would you call that ground tissue, and why wouldn't you define it? And it's just the nature of how our profession is. You start picking up a word and then it sits there. And there's some wonderful words. I mean, we've got lots of very precise words, but we also misuse them. Callus and woundwood. There's another one that, the tree is healing, the tree is occluding. Just slight differences in the word convey very different things. So hopefully that answers that one for you, Niels.
Yeah, no, that's good. That cleared up because I was wondering. I read that, so I was going to ask you, and then you brought it up. It's good because I forgot.
Yep, it's good. I forget everything all the time.
Thank you.
So the next one, then I need to go to emails to share those. It's the one thing about not using an infinity tab, Peter. I might just try it again.
Yeah, just try
It will go to infinity,
We
but
can-
I'll take it off when I go to something else. I understand why it doesn't like it. I do make it do things that it shouldn't do. But okay, can you see my
Yeah.
Okay, great. You can see my emails. So another one there is a question about injections, and I will come back to that, and we will answer that really easily within this evening. But I thought I'd go to an earlier one that was sent through, and this one was from Joe Floyd. I don't know whether Joe's on the line, but he'll pick it up on the recording. And here's some cypress that aren't doing particularly well. Now, let me see if I can get some more information from, Joe is based in Sydney, so now I know a whole pile of information. Cupressus of some description, looks like sempervirens, but I'm not prepared to bet my family home on it. Fairly big cone, so I'm guessing it is. And the trees are not looking good. Does anyone want to have a first guess at what the issue here would be? What do you think it might be?
I've heard of weevil or some such that gets them. I don't experience them much myself.
Okay, yeah, you don't get them very often, Peter, because you've got fairly good growing conditions.
We don't have a lot of those cypress around here either.
Yeah, and that's probably a double win. But cypress weevil is a native beetle, native Kirkleonidae, that does cause problems, but it's pretty obvious to pick up. You'll end up with chewed tips, which I can't show you here, but I can show you images of, and then round holes. And I don't see any round holes in that bark. So the bark looks fairly entire.
Could it be ground disturbance?
It could be. How would you describe Sydney at the moment, Vicky?
Well, it's overcast, very humid.
Okay, humid. Are we? What are soil moisture levels like?
Well, it's been dry. We've been in drought.
Yeah, we are in a drought. There's no questions. You drive around, you see a whole pile of drought response in trees. So we're in abnormally dry conditions. We typically in Sydney get a winter, particularly in the western part, we get a wet winter and a somewhat dryish summer with thunderstorms mostly. Well, we're way below on our moisture levels. And of course, that's on the tail end of 2 years of good growing conditions. So now we're in a bushfire risk situation. So that's one thing is the dry weather. So assuming that these photographs have been taken the last month or so, I'm now starting to get my once or twice a week calls from people saying, hey, my cypress is starting to die. You know, I'm wondering whether someone's poisoned. I'm wondering whether something's happened. Second thing is, this is in a hedgerow. And if you remember the story of the hedgerow, when these were young trees, they were competing against each other for the soil. Now they're bigger trees, they're competing for each other, and often in urban areas for exactly the same volume of soil. And so proportionally, our amount of moisture has dropped. The tree's response, inefficient branches are shed, internal tissue browns off and dies off and is got rid of because it's able to photosynthesize less effectively. So we get thinning on the inside of our cypress hedges. And so I can assure you, if you're Sydney based at the moment, or if you're Melbourne based, you're probably going to be finding cypress hedges not looking quite what they were 2 years ago. So that's my gut level without Joe being able to give me any more information. There's a little bit of resin. Now we know what that is. It's a protective layer. Nothing like you'd expect from either cypress canker, Ceridium, or from cypress weevil. So perfectly within the range of normality. Why the splits? Don't know why that one split. That looks like it actually is a split, but a lot of this is just growth activity. So as the tree grows, it stretches, the outer bark gives way, and we end up with a rougher bark, which is why you end up with something like a willow, which is when it's young, has got a beautiful green, smooth bark. And by the time it's, 10 or 15 years old, it's got a really rough bark. It's because the growth is in that cambium zone, causes stretching on the outside cracks. Things like smooth bark trees, they produce a bark cambium that allows them to shed out an outer bark every year. And so they end up not with that split and furrowed look. So that's our smooth bark trees. So that's that question. I think, Peter, that was the only one apart from the
yeah, I think so.
Yep. So apart from the question about controlling insects, and I'll come back to you guys on that one. If I can, if it'll let me. It'll eventually let me. There it is. Now it has. Okay. So controlling any insect involves a chemical. Mostly. There are a few that don't. So, for example, cup moth, there's been this discussion, hi Hank, there's been this discussion about using water, for example. Just by spraying the foliage, you end up increasing the natural mortality rate of something like a cup moth, or at least its larvae. But most things involve a chemical. Now, when you've got a chemical, Hank is from Queensland.
Correct,
Different
Mark.
rules.
Yep.
Different rules. Somewhere around, Mr. Frey was on earlier on. There's someone there. Let me see. Mark. Mark is from Tasmania. Different rules. Victoria. David, you're from Victoria, aren't you? Run. You get it really easy. Hank gets it easy. If you're in Sydney or Tasmania, South Australia, Western Australia, I'm not sure on. I just pretend that doesn't exist. You end up with a whole pile of different requirements. In New South Wales, I need to follow the label or apply for an off-label permit. In Victoria, you need to be able to show efficacy, that is, show that the treatment is acceptable, it works, and have insurance for doing it commercially. Hank, I'm pretty sure you've got the same requirements in Queensland, which is this very broad saying, okay, it's ridiculous to get out a permit for everything. If you're covered against it and you screw up, then we get to sue you. So as each state varies, I'd say talk to your local authority just to make sure. But controlling insects is a fairly easy process in terms of injections, provided that our chemical is water soluble, okay, or can be slowly released. So we can bind it up, for example, a chemical up in a polymer or in a glycol, and make it go into the tree very, very slowly, but continuously over, weeks and days and months, and sometimes a year or more. If we want it to go in quickly, we want it to go through that tube system, the apoplastic system. So we want to get into the vessels or the tracheids. Then we've got to understand how that system works. So it's got to be water soluble. If you want to put it through tracheids and they've got bordered pits, they're little plugs that block up the tracheids, forget it. So trying to inject into something like a pinus, you're not going to be able to get free flowing fluids in through a pinus. On the other hand, if you want to do the same thing to a Taxodium distinctum, no troubles. Go straight for it. It doesn't have these bordered pits. It's got simple pits and it'll move through. Slower than it would through, say, something like a eucalypt, because the tubes are smaller. And the smaller the tubes, the slower things move. Okay? And it's the same thing if we apply it to a chemical to the soil. There are chemicals that, for example, with the elm leaf beetle, you can use a drench. You can drench around the trunk of the tree, out a little bit, but essentially near the trunk. And it gets taken up by the root system and transported. So that's a really nice systemic process. And the most common chemical used for that is imidacloprid, Confidor, and you can buy that. Mallet, I think, is one of them that's approved for soil drench on elms for elm leaf beetle. One of the nice things about soil application is you don't damage the tree. Don't damage the tree, and the second advantage of it is you get a long uptake. So you get this treatment that will often give you a couple of years rather than 1 year. And oftentimes these new diseases that you get outbreaks of, and David, you'd certainly see it with elm leaf beetle down your end of the world. For many years you just get an infection, you sit there and think, oh, they're a pain in the butt, they've ruined the leaves, but they're not at high levels and they're tolerable. And you get the cyclical outbreaks. And they're the problem. Then nature comes back in and starts to control that back down. And we saw it with Chevaphus celti, the woolly hackberry aphid, came into Australia through Richmond. I won't blame the RAF base, but you can figure that one out. We saw it for the first 3 or 4 years. It was horrendous. There was just so much honeydew produced. You would see puddles of it under trees. That's how bad it was. And we thought, oh, this is just vile. Now our natural predators have seemed to have brought that back down under control, and we've just got a balance. It's not that it's not there. It's not that it doesn't occasionally do things, but it's not like it was. We found the same thing with elm leaf beetle when it hit Sydney. Conditions weren't as ideal for it as Melbourne, and every now and again you'll see one that gets to a bad proportion, but generally it's controlled. And Vicky's in Sydney. Vicky, how many times have you seen a tree 50% stripped, an elm tree, by elm leaf beetle?
Yeah, I haven't seen many at all.
Yeah. So whereas David is a little bit more frequent in your end of the world because your environment comes closer, and that's that thing we've talked about before called the disease triangle. We've got a host, we've got our disease, and we've got the environment. And if we can skew one of those either which way, we can change the way that disease performs. And so in Melbourne, you've got slight advantages. You've got a lot more elms. That's one of the skewing factors. So the habitat's more ideal. And I suspect climatically, it's probably a little bit better than we've got. Hank, you've got Oh, lantana flatted. Lantana not flatted. Yeah, lantana flat. No, leafhopper. Lantana leafhopper. Great little creature. And it really hammers fiddlewood. Really does. Just knock the hell out of them. If you're near the coast, Andrew, you're up near the coast for Sydney.
No, I'm in Melbourne actually, so elm leaf beetle's pretty bad down here. Yeah.
Yeah. Okay. So the lantana leafhopper, great for controlling lantana, destroys fiddlewoods, will kill them, but you don't see it anything west of Parramatta, because just one hot day of 38 or more, and you'll just start seeing them drop out of the tree. And so these are the things we're going to deal with. Okay, so how do you get things in and what do you do? First of all, you want to always use chemicals correctly. A little bit is good. The right amount is ideal. Adding more doesn't make it good. Okay, and there's a real tendency to say, look, I've got a big problem, so let me double the chemical. Resist and avoid the temptation. I know what it's like, but the efficacy figures that you get, it'll be whatever it might be, 4 grams per 100 mils of Trunctoma, or whatever the chemical you're using is what you should follow. So let's say we wanted to inject it, and we're going to use a water soluble. There's some really great water soluble insecticides. Probably the most ideal in my mind is abamectin. It's a bacterial extract, so it's got a fairly big LD50, means it's got low toxicity to people. It's water soluble and fairly easy to get into a tree. How would you get that in? Drill a small hole, and then with just a little bit of pressure, put it in. If the tree is photosynthesizing, you can get rid of 20 mils of mixture in 3, 4, 5 minutes. On a cool day, half the canopy is gone because you've had the elm leaf beetle come through. It might take you 30 minutes to get that same injector emptied. And it depends on the injector you use. There's some really good ones out there. Proud down out of Melbourne that have a really neat glass one that's got a full application system or closed cycle. You can buy ChemJet ones that are, I don't know, $15, $20 a piece. And they've just got a spring and they're cleanable.
Right.
And you batch up whatever you want into those. There's all sorts of different injection material, but tree injection is so much smaller industry here in Australia than it is in America. In America, massive, big, multi-million dollar industry. And there's some reasons for that. One is they've got far more exotic problems. So, for example, they've had emerald ash borer. It's a Buprestidae, so a jewel beetle. It's been a real problem for them. And so they want to try and get rid of it, or at least control it, manage it. They'll never eradicate it now. And that whole process of chemicals in trees has been a part of the American culture for over 100 years. So, and particularly when codling moth, which is a flightless moth, came in, there was a desire to try and eradicate. And guys would go out with, and this is my lifetime, chemicals like chlorpyrifos, chlordane, heptachlor, and spray. And you think, those chemicals were just plain vile. And so they've come from a really chemically focused history. We've come from a non-chemical history. So the harder thing here is to actually say, yes, I'm going to do it. I'm going to inject. I really do need to intervene. And so that's the harder action for us to take. And again, just a show of hands, who has done an injection of some description in the last 12 months? Hands or yay?
No.
Yeah, you can see he's from Queensland.
Yep.
And Neil's. Okay, so that gives you an idea. It's not the norm. And then the next thing is, we're consultants. We want to get paid for consulting, and all of a sudden I'm doing something like that. I charge them consultants' fees on top of recovering my cost for the chemicals and recovering my cost for the time that I'm going to spend doing it. It becomes a difficult process, as opposed to the tree company that already has a spray rig or a fertilization unit or something like that, which is typical in America. Probably 10% of all tree companies have a spray rig. Just out of curiosity, who has a petrol or diesel powered spray rig?
I've got a battery powered one.
Doesn't count, Peter. Does not count. So yeah, and that's just, again, we don't have that history of doing things. And our biodiversity, in spite of all those things, has done an amazing job. You think about it, we talk about Lantana flathead or leafhopper, Lantana leafhopper. It's really not done badly at all. You think about what else have we had come in? Elm leaf beetle. Okay, well, it's done a bit of a knockback on the elms, but elms are still a major feature tree in Melbourne, I'm assuming, David?
Absolutely. And yeah, where I am in Mansfield, they line the streets. So yeah.
Yeah, so really hasn't had a huge impact. You do some chemical treatment down, or the companies do. There are 2 or 3 big players that, turning over several $1 million a year. And then a whole pile of less size ones. Most people with a spray rig. Hank, your biggest one would be Adam.
Colin Curtis.
I'm assuming. Let me kill Colin Curtis. Your biggest one would be Adam in Queensland, and his spray rig doesn't go out all the time. I know that.
Yep.
He sits in the shed 4 out of 5 days at least. So yeah. How do you find out what you're going to use? Again, depending on where you are, you're ideally looking for an already approved permit, a minor use permit. And minor use permits are weird things. They're hidden away in the archives of almost nowhere at the APVMA. The APVMA is the regulatory body. They tell you whether a chemical can be used, how it's going to be used, what's got to be on the label, and all those things. There are some weird and wonderful minor use permits for things that are just for a short term. We've got a problem with something like Lantana leafhopper. Oh yes, well, you can use imidacloprid as a spray for that. Great, okay. Or you could use I'm just trying to think one that's just come out. In Western Australia, they've just had a big problem with polyphagous shot hole borer, a new exotic disease that is destined to hit the East Coast, but they're doing somewhat of a questionable job of controlling it. I don't know how they're going to go longer term, but I think we can all expect it over our end of the world. You can kiss maples goodbye at that point in time. Sorry about that, David. There goes some more of your trees.
Absolutely.
And they are, and they're very exposed because these, the case of polyphagous shot hole borer, it likes trees with low levels of what we call extractives, or that sapwood that just stays pale and doesn't put a lot of chemicals into it. So yes, there's an off-label permit or a minor use permit issued to control polyphagous shot hole borer. And again, I think it's an abamectin based spray and injection in that one. And so how do you find out the dosage? Well, look for similar insects. So if you're talking about Hemiptera, they're going to tend to be the same application rate right through. Lepidoptera, different than Hemiptera, Coleoptera. So each of your groups, each of your families will require a different approach. So we talked about Confidor. Confidor is a wonderful chemical, imidacloprid. It's a neonicotinoid, so it's something like a nicotine molecule that's been twisted. It's very safe for mammals. It's very dangerous for aquatics and for insects. But oddly enough, it's not really effective against Lepidoptera. So spinosads, on the other hand, really effective against Lepidoptera. And Bacillus thuringiensis, very, very effective, as long as you can get it between the second and the third, preferably at the beginning of the second instar. It's a natural biological control. The only problem with that is if it rains, you really need to do it again. It needs to eat the spore in order to be effective. But, it's one of those wonderful chemicals that, unless you happen to be a Lepidoptera, you're pretty safe to have it sprayed on yourself. I'm not saying you should ever spray a chemical on yourself, but it's the chemicals are well adapted to being very specific. In that case, it's not a chemical, it's a bacteria. So now injections, the critical thing is you need to understand how the inside of the tree works. That is, you need to know whether it's got vessels or tracheids. So gymnosperms have got tracheids. If it's got tracheids, you need to know, are they simple or are they complex? So they've got bordered pits or are they simple pits? And that's going to have some effect. You need to know whether the chemical can be taken up through the soil, and something like imidacloprid, as you've gathered on the label, it tells you it will be taken up through the soil. So that tends to be a pretty neat chemical. The drawback, it's an insecticide. It's not selective. So it's going to affect other things in the tree as well. And you put it in the soil, it's going to affect things in the soil. And that's got good and that's got bad to it. There's nothing's absolute in any of this equation. So it's going to kill off soil organisms. And there are things in the soil that aren't good. For example, we've talked about this before, cicada nymphs. At the moment, I've just had my Pakistan mulberry raided by the red-shouldered beetle, plague beetle, Monoleptera australis, and just hammered. And we must have had probably 1 million of them on the tree. I know that next season I'm going to have a doozer because they're all now laid eggs. The eggs have hatched. We've got larvae now in the soil going around, and all of a sudden next time around I'm going to have 100 million beetles. So, you know, sometimes those treatments in the soil have some unintended beneficial consequences. Sometimes they've got unintended negative consequences. You start affecting things like earthworms and things like that. Spray is great, but you've got to be able to get there. And something about putting in most insecticides up, 15 meters or 10 meters from the end of your nozzle is not something that's generally encouraged. It's not that you can't do it. You'd have to think about it. So if I was spraying with Bacillus thuringiensis for Dipel, is the trade name here. If I was spraying with Dipel for an outbreak of, trying to think, brown-tailed mistletoe moth, horribly itchy, irritating caterpillar larvae. If I was spraying for that on mistletoe on a tree, I wouldn't have a problem with it overspraying and some of it landing in the pool next door, because unless those kids are absolute grubs, they're not going to get killed by it. Okay, so it's so non-risk to things apart from Lepidoptera. But I don't know that I'd want to be spraying or doing a soil injection where somebody, for example, was harvesting groundwater. That would tend to be something that I'd say, well, hang on. Or I've got shallow rock shelves and lots of cross flow. There would be times when you've got to say, hey, spraying is probably the better option there, or stem injection is a better option. So I don't think there's ever one simple answer for all those things. Almost all the chemicals that you'll see used overseas or where there's research done, almost all those chemicals are licensed in Australia for some use. Dealing with the APVMA is, I'll say, laborious. So someone like Hank can go and relax and doesn't have to worry because he just, that's fine. I had the Forestry Commission in Tasmania call me a number of years ago and said, Mark, this is just crazy. How do we get past this problem? It's just destroying their research stuff. They couldn't get permits just to even do research. So it really is situation specific. And the ARB Australia has talked about it, and Peter and I have had this discussion on a few occasions about going ahead and trying to get a limited use permit for urban trees for several chemicals, abamectin and imidacloprid being the two main ones. And one day we might pull the finger out again. The last time I did it, it took me 3 years. And I won't mention the name of the chemical company, but we had a big chemical company, starts with B, that spent a lot of time trying to stop us from getting the permit. Because again, there's limited amount of money that can be made in the tree industry, and you want to try and get a good return for getting a label for something like Monsanto might cost them $40,000 or $50,000 to get, and so they've got to sell enough product to get that money back. And so they tend not to want to have these minimum and off-label permits freely available. And the last one we had, I think we had for 3 years an opportunity to use imidacloprid as a stem injection and a soil injection, provided you held used chemicals, the ChemCert certificate, and the AQF level 5 unit in diagnosed tree disease. So, yeah, if we were to go through that process again, would anyone be interested in us looking at doing that? Yeah, Melbourneites. Yeah, I know you don't care, Hank. I get the deal. Don't rub it in. So 2 there said, yeah, they'd be interested. So that's something we'll keep in mind, Peter.
Yep.
So any other questions on injections? A few other things. Trees take up best when the soil's moist and has been for a few weeks, so you've had time for absorbing roots to get going and function, because all of these things have to be moved around. If you're doing a soil injection or a stem injection, things have to move. I've seen a healthy young sapling, eucalyptus sapling, take up 20 mils in less than a minute. Just as quick as that spring can push back in, it's gone up the tree. And I've seen people try and put things in that shouldn't go in. So a couple of the ones that do come out in that was Pandanus flatted. Was a problem in Queensland and southeastern Queensland and northern New South Wales, and there was a real push to try and get rid of it. And a lot of people were doing injections into Pandanus. Now, Pandanus are vascular bundles. And so what was happening is they'd put their injector in, they'd use something like a Sidewinder, which is a pressurized system, push it up to 120 psi, because at about 105 to 110 psi, we start to get tissue damage, and they were just blasting the cells to death, pressurizing them. Okay. And I've seen that on more than one occasion where the pressure's been pushed up. So generally, if you're much more than about 20 or 30 psi, you really are forcing the system. You shouldn't be doing it. So some things don't go in. So if it's not water soluble, if it's something like a wettable powder, okay, and I'll tell you, it'll say WP on the label. It'll say Confidor WP, wettable powder. You can try and stick finely ground up chalk through holes that are about 10 times smaller, but it's not going to work. And that's the process of putting things like that in. And I will mention young Sean Talent, because he's always good to us to learn from. Young Sean Talent tried to do that with some Confidor. Confidor's the wettable powder, the Silver Shield. It's broken down, I believe, into a glycol subbase, and so it's a different mode of action. But wettable powders just do not inject into trees. The blocks up the system, and that's it. And then that's when people start pumping and humping and then saying, "Oh, it didn't work." Happy with that? I'm not going to tell any of you exactly what to do or how to do it, because then I become equally party to it. But I can tell you that there's plenty of information out there. Just look up, efficacy, imidacloprid, soil injection, efficacy, abamectin, stem injection, those sort of things. And you'll find, particularly in Google Scholar, you'll find papers eventually. You might have to put 3 or 4 hours into it. But then I'm not saying any of you would go do it off. David, you can do it off label. What am I talking about? Andrew, you can do yours off label. You just go ahead and do it. Make sure you are insured for that risk. And my understanding, that's the current requirement. Happy with that?
Fantastic. Thank you.
Okay. No, I know it's not a perfect look. If I was going out there, and I was only doing it occasionally, I'd go out and buy a Silver Shield kit and spend a couple of $1,000 and wait to get my money back over the next sort of 4 or 5 years. If I wanted to do it on the cheap, I'd go out and buy myself some ChemJets and look for alternatives rather than Silver Shield. But in terms of ease, it's already set up. It's already a nice system. It's already got, but again, limited permit. If you wanted to go control Monoleptera australis, the little red shoulder beetle that I got the other day, does everyone know what that looks like? No.
Yep.
Let me. I'll do an entire screen again. I apologize, but it's probably the quickest way. Share and go over to here. Yeah, Peter, you'll be familiar. It's quite common up your end of the world. May not like that. No. Red. Okay, that's it there. So it's Chris Miller Day. That means it's one of the leaf-eating beetles. And they do a fair amount of damage, and they come out not just in a few, but in massive numbers. A big pest of avocados. One of the spots where Peter will come across it. Big pest of flowers. So again, on the macadamias, they can be problematic. But pretty neat little creature. Apart from that, and I've certainly seen them here in plagues of probably 5 to 10 million in a single thing, and it goes from 0 to those sort of numbers in a matter of 24 hours or so, and they just go through and they strip everything. You get them occasionally up in your end of the world, Hank. They do a nice job, particularly again in avocado. I know it's a problem for them. So, yeah, neat little beetle otherwise. Now, any other questions?
No.
Guys, next tips is no longer going to be tips. Next tips is war. That is, next week, war is not on. I think Peter sent out to you about that. I'm in Melbourne. Those of you coming down to play tree protection with me on the Monday, I figured after speaking for 8 hours, I should try and preserve my voice. For the first time ever, last presentation I gave, I ended up with throat strain and had steroids to be able to go back to breathing normally again. That's unusual. Never had it happen. So I'm just taking it a little easier on my voice. So 8 hours of teaching, I figure that's probably pushing it enough for the day without doing war afterwards. So our December meeting will be war and not tips, so that we've had equal number of tips and war throughout the year.
And of course, the last Monday of the month in December is the 25th. So,
and I figured none of you would want to join me, which is really, really sad. But yeah, if you do want to join me on Christmas evening to discuss Christmas trees, I'm happy to do that. But yeah, so this will be our last tips for the year. In the meantime, in the next couple of months, insects tend to run rife in this end of the world. Our time of the year is really odd. Insects tend to be at their highest. Diseases tend to be at the highest sort of from spring until autumn. So if you've got a question in the meantime, flick it across. We will answer it when we come back in January, and we will be back in January for tips like normal.
Hey, Mark,
Yes.
if this is the last tip for the year, I do have another question, if that's okay.
Yeah, please go for it.
So I was just wondering about salinity and trees. I don't know if you get much of that up your end, but there's definitely a fair bit of it here along the Murray and northern central Victoria.
Yeah. No,
my partner's actually working.
We don't.
Salinity up there.
I was fortunate enough to go hear a lecture by Kim Coder on salinity, and I know that because Kim's given that lecture, the paper and all the supporting material is available on his website. And Kim is a brilliant research scientist.
What was his name?
Kim Coder, Coder. And
Yep.
he's at Warnell School of Forestry and University of Georgia. It's the same end of the world, same place. The issue of salinity is really complex. And he was looking at salinity from storm surges, which have been a big problem in coastal areas of the US. And a lot of it depends on where you're at. So that your big problem is how to get rid of the salinity once it starts. And this is the flushing process that's incredibly difficult when you're bringing the salinity up from below. And one of the things that we do know that from the very experience of where you are was that removing trees was a dumb thing to do.
Yes.
And irrigating was even dumber. Because that tended to let our deeper salts that had been washed down start to work their way back up to the surface. And
we're apparently seeing pretty bad salinity levels at the moment up in north central Victoria. Apparently the worst since the 80s.
Yeah.
We had so much rain for 2 years, and now it's really dry.
Yeah, and so it doesn't work the way you want things to work. You can see how you think that adding water solves the problem, but adding water allows deeper salts to be brought back into solution and brought back up. And so, particularly when you've got evaporation, it's really good at bringing it up through the soil. So, yeah, I don't. It's really not in my field of expertise, apart from knowing that small amount about it. We don't use desalting processes, de-icing processes and salt. Sodium chloride is a nasty chemical. It does interfere with a lot of the biological processes. And then there are plants that are just so brilliantly adapted to them. One of them's a large number of the eucalyptus species, for example, are well adapted. In fact, some eucalypt species can handle being irrigated with marine water, ocean water.
Sugar gums are pretty tolerant of salt.
Yeah, so I mean, the thing that I would be looking at doing if I had to grow in an area where sodium was an issue would be looking at growing species that were better adapted to it. And
I do recall seeing something, whether it was something on TV or whether I heard it some years ago about using saltbush to combat the effects of surface salt.
Yeah.
Basically, any plant makes better.
Yeah, any plant that's salt tolerant, and there's a heap of them, and there's a heap of Australian plants because we have large areas inland where we do have higher salt levels. For example, our salt pans of the inland. Working with those, and of course the problem is the other half of it, of course, is a lot of the water along the catchment where you are has been used for irrigation, and that just exacerbates the problem.
They do flood irrigation up there, which is probably the worst for it.
Yeah. So I don't think there's, apart from trying to change practices, I know there's been a push never to do the same thing again. I think when the Ord River scheme went, it was a little bit wiser. But yes, we certainly have made a real stuff up of the Murrumbidgee Murray combination in terms of our management of water. And it's a difficult one because we're very different, and I don't know whether you've ever thought about it, we're different than the US. The US gets this stuff called snow. We get little sprinklings of it, but they get lots of it, and that means their snow melt is really valuable to their management of their land. We've tried to do similar things. We put this nice big dam in, but the process hasn't been as effective because of the way that the way that our land is. So yeah, we're probably not as well suited to mass irrigation as other places.
We rely. I think the Dokielka Mountains, or the mountains of New South Wales, isn't that where the the Murray River starts, really.
Yeah, yeah. And so our snow melt is important for the flow, and then we intercepted it for hydroelectricity, and now we're intercepting it again for, well, we say we're going to do it. I don't know whether they're ever going to finish building it, doing it to use temporary energy storage, pump it back up, and then let it come back down again. You know, I watched a doco today on what's happening with old wind farms, and that's plain scary. So I'm not sure that we're any closer to solving the problem, except I can tell you that if you want to sequester carbon, trees are still the number one. If you want to help regulate temperature, trees are still number one. If you want to improve human health, trees are up there at number one. So we really, our solution is still the same in terms of trees. It's just picking the right ones when we've come in and screwed things up. You know, but there's still plenty I think you can have, Niels, in terms of good species down the other end of the world, but I wouldn't want to go run a 5,000 acre almond farm. And of course, hundreds of, well, not hundreds, but certainly there are several that are in that category down that end of the world. If you haven't been down there, guys, it's worth the trip down there, isn't it, Niels, just to see the scale of some of those farms. Mine,
That's pretty crazy.
yeah. Yep. Okay, guys. Well, I'll see some of you